Ok, buenas tardes a todos. Es un placer para mí dar inicio a esta segunda sesión del ciclo de charlas especiales de la reunión de la Asociación Física Argentina sobre el COVID-19. Es un problema que nos tiene en vilo a todos. Hoy tendremos las últimas dos charlas, mejor dicho, plenarias del ciclo. Junto con Karina Laneri queremos agradecer a los oradores por su gentil participación y también a la comisión organizadora por el apoyo para realizar esta sesión especial. En particular queremos agradecerles por el esfuerzo en lograr que estas charlas se retransmitan en vivo por streaming en YouTube y sean abiertas para todo público. Si quieren divulgar el link de streaming, lo estamos compartiendo en el chat de la sesión. Eso sería para los socios de la AFA. Karina, está silenciada.
Gracias, Gustavo. Together with Gustavo Sibona, we want to thank the speakers for accepting this invitation. The first of the talks will be in English, but you can ask questions in the public chat here or in the web, also in the web chat. If you prefer, if you feel more comfortable, you can do it in Spanish, and then we will translate them after the talk to Professor Saveez. Our first speaker will be Professor Saveez Saffarian from the University of Utah in the United States. He's professor of physics and biological sciences. His lab is an experimental virology and biophysics lab that develops microscopy techniques, merging live imaging with high-resolution microscopy to visualize viruses like HIV. And he was recently awarded by the National Science Foundation to study the structure of SARS-CoV-2. So thank you very much, Saveez, for sharing your research with us.
Thank you so much, Karina, and the organizing committee for the invitation. It has been really great to have this opportunity to virtually be with you guys in Argentina. I always love to visit that country. I wanted to just kind of briefly tell you where we are. We are in Utah. Here, if you can see, this is the red state, somewhere in the western United States near the mountains. These are the Rocky Mountains here, and as you can see, we have a lot of skiing, a lot of hiking, and a reasonably big city. So I really welcome you to write to me and to keep in touch after this presentation is over. So today I'm going to be talking to you about looking under the hood of SARS-CoV-2, which is the current agent that is behind the COVID-19 disease. I'm coming from here, this lab. My lab is kind of highlighted here at the foot of the Wasatch Mountain. And a lot of what I'm going to be talking to you about today is about a really, really close collaboration between myself and my really good friend, Professor Michael Vershinin. Michael and I joined the University of Utah about 10 years ago together as assistant professors. Both of us were interested in biophysics, but I was a lot more interested in viruses. And Michael comes from the group of, he did his PhD in the group of Ali Yazdani. So I think the condensed matter people in the audience would probably know that lineage of physics. Michael specializes on a lot of surface scanning technologies. And in his lab, he has developed a lot of optical trapping. Then he looks at molecular motors. And so we have gone really close. And after during the COVID, we have been having a really active, fast collaboration to try to look at this virus. So in the outline, I'm going to basically introduce you to pathological viruses. I think that this is the Argentinian Physical Society. And if you are like any other physicist in the world, you probably heard of the virus, but you don't know much about what viruses are. So the first part of the talk would be an introduction to viruses. Then we are going to zone into coronaviruses and specifically SARS-CoV-2. And at that point, I'm just going to make a case for a rapid research on SARS-CoV-2. So what are the avenues in which we can contribute as physicists to kind of look at SARS-CoV-2 and what we can learn. I'll tell you a little bit about production of SARS-CoV-2 virus-like particles that has been going on in Utah for the past almost a year now. And then the temperature studies of looking at structural stability of SARS-CoV-2. So there are overall 21 virus families that infect humans. And as you can see, they are named mostly according to how they look like. For example, this viral family here, the rhabdoviridi, is called the rhabdovirus because the viruses in this family look like bullets. And you can have the rabies virus is part of this, the VSC, which is a very common livestock virus, is part of this. Filoviruses are elongated viruses, so they look kind of like a filopodia. So a lot of the virus naming has been coming around from how the viruses look like. And I think that you would not recognize any of these names specifically because these are names of general viral families. But you would recognize the rogue agents within these families. For example, within the whole retrovirus family, you have HIV-1 that is a very specific retrovirus, but it is a very potent human pathogen. So is the same thing with the orthomyxoviruses. There are a lot of different orthomyxoviruses, but one of them is the flu virus that we know. And within the filoviruses, there is Ebola. And so coronaviruses are the same. There are a lot of coronaviruses because they look the same. They have the coronas on their surface. That's why they're not named coronaviruses. But there is only one SARS-CoV-2, so it's a very specific coronavirus that is now being infectious. The question that we get a lot is, well, have coronaviruses been around before? And I'm pretty sure that any one of you have been probably infected with the coronavirus before, may have been infected with the coronavirus before, because they are common cold viruses as well. So as I said, these large family viral tree really is mostly governed by how viruses look like. And I specifically want to tell you a little bit about how virus shape and virus function are uncoupled a little bit, and what kind of how much it took us virologists to kind of understand that. It really took a long time. So viruses were not known pathogens until the advent of electron microscopy, because viruses don't grow by themselves. So they really do not follow the core principle that was established for pathogenic reagents. And it really took electron microscopy to visualize them. So that's why the first set of viral characteristics are always visual based on how the viruses look like in the electron microscope. But what do really viruses do when they come into your body? In order to understand this in a very general sense, we really need to kind of go back and understand how the body deals with genetic materials, because the virus is really a bullet full of genetic material that needs to replicate. And so if you want to understand how the virus works, you really need to have an understanding of how human cells work, or how eukaryotic cells work in general. So here, I want to focus your attention to what was developed from the 50s in our understanding of how genetic material gets translated and transcribed and takes over the biological function. So all the genetic material you have is encoded in DNA as part of genes. But DNA is like cooked spaghetti. It's as specific as cooked spaghetti. It has no specificity. It doesn't really do. It has no chemistry involved in it. It doesn't really do anything. So in order for chemistry to happen, in order for things to actually start working, you need things that are chemically active. And so the first thing that happens in the body is that the RNA polymerase would read these DNA, the genes in the DNA, and create a messenger RNA. Now, messenger RNA molecules are very similar to DNA, but they are a lot more chemically active, and they're a lot more short-lived. And then the mRNA by itself, it's speculated that there exists an RNA world long time ago, before proteins came along. And this was basically the basic premise of life, but I don't want to take you there right now. In your body at the moment, every time a gene is read into mRNA, that mRNA is captured by a ribosome. And ribosome is this massive enzyme that would take the RNA and create a protein out of it. So this is what's going on in our body constantly. So we have polymerases read our genes and then create RNA. RNA is read by the ribosome and proteins are made. And proteins are really the ones that respond to the environment. So whatever you're doing is because some protein is synthesized somewhere in your body. Of course, I'm doing a spherical approximation of the cow here. There's a lot more complex things going around in your body, but this is one of the overarching themes. So how do viruses interact with this? Viruses are amazing because viruses do not have the ribosome. So they have genetic material and they are a lot more free. So they're a lot more loosey-goosey with their genomes. So when you look at your genetic material is protected and error-proof constantly so that you do not develop massive mutations that would lead to cancer. Viruses, on the other hand, are just very loose with their genomes, and they have viral enzymes that then read these genomes and create mRNA. So the whole purpose of the virus is enter the cell, inject its viral genome into the cell, have its own viral enzymes read that genetic material and create mRNA. Once you get to the mRNA level, the cell does not distinguish between the mRNA from the virus and its own, although that's also a simplification. And then they take over the ribosome and create a lot of protein. So they basically shortcut, they short circuit the central dogma by hijacking the ribosome. So given this, that now we know about viruses, in the 60s and 70s, David Baltimore, who was one of the really good virologists, he thought that the viral families are a little it's a little not well characterized. So if you consider, for example, the negative strand RNA viruses, they're all different shapes, but they share exactly the same genomes. And so David Baltimore reorganized the viruses into 7 different viral genomes that require certain viral enzymes to replicate within themselves. And as you can see, coronaviruses, which are highlighted here in red, in electron microscopy look like this, and they are positive sense RNA viruses. And they actually, when they get into the cell, they create mRNA and take over the cell. So this is just kind of like a general overview of viral pathogens and how they interact with the body. And now I want to kind of focus in and zoom into the actual structure of the coronaviruses. These components are shared between all coronaviruses. The coronaviruses are polymorphic particles, which means that their shapes are not identical. They have a size between 120 to 160 nanometers, so their shape size varies. They are enveloped, so they have a lipid envelope that they hijacked from the host cell. Within this envelope, they have a membrane protein, and many copies of this membrane protein are inserted into the envelope of the coronavirus. So is the spike glycoprotein. And in combination, the spike glycoprotein, the membrane protein, and this protein here in blue, that is the E protein, are the structural component of the coronavirus. So if you think about the viral family, the structure is independent of the genome in a way. What gives the coronaviruses their shape and the structure is really these 3 proteins that create the viral particle that then encapsulates the genome. The genome itself in the coronaviruses is extremely complicated. So if you look at the genome of the coronavirus, first of all, it's extremely large. So the coronavirus genome is about 30 kilobases, which is about 3 times the size of the HIV genome. And when you have the virus has these spikes on it. It comes and binds the surface of the cell. So this can be basically a viral particle that was floating around in the air. And you inhale this particle, it comes in, and it attaches to these ACE2 receptors that are sitting on the surface of the cells at the back of your throat, for example. The virus sits around on ACE2, and then it internalizes and the ACE2 receptor kind of brings it into an endosomal unit. Although this particular step has not been completely visualized for SARS-CoV-2, but we believe that that's what's going on. And then these S proteins, these spike proteins, actually mediate the fusion of the viral membrane with this endosomal membrane. And once this fusion happens, then the viral genome is released into the cytosol. So this is where the virus would be basically hijacking the central dogma machinery of the cell and creating a bunch of mRNA. And that's why, the understanding all the production of transcripts that happen in this stage is very important, because each one of these mRNAs, when they're transcribed into a protein, translated into proteins, their job is to take over something in the cell. So in a very short amount of time, the cellular machinery is completely taken over. If you want to think about it as a non-equilibrium process, really the whole chemistry inside the cell has been changed by all of these proteins that the SARS-CoV genome has expressed inside the cytosol. As far as structure is concerned, the genome transcribes the S protein, which is the surface protein, the spike protein, the M protein, and the E protein of the virus. And these proteins assemble on these internal vesicle called the ergi, which is somewhere between the ER of the cell and the Golgi. They assemble on this surface and they bud into another vesicle. So the virus release really happens inside the cell. It's as if the virus is releasing into the internal vesicles of the cell. These internal vesicles are then moved to the surface, and new progeny viruses are released back into the environment. So every virus gives away about 10,000 to 100,000 progeny virions, and these are the viruses that then go in and infect other cells. So this is the kind of the life cycle of the coronavirus infection. Now, this infection actually requires, in order for it to go from host to host, it requires a particular delivery mechanism. And the SARS-CoV-2 virus utilizes the delivery mechanism of the liquid droplets. So if somebody who is infected with the virus, their upper respiratory systems are producing tons of virus. Imagine every cell in the upper respiratory system producing thousands to 10,000 viral particles. So when this person sneezes, then these viral particles are coming out of the mouth and nose of this infected person, and they create this flow of viral particles that then kind of goes out. And at some point, all of these, because there is a lot of small droplets and a lot of air involved, the surface area of the droplets is really large compared to their volume, these droplets dry out. And so there is a lot of kind of dried out virus particles that are deposited on the surface after somebody who has sneezed is left. So these viral particles, as I said, have this S protein, which is the main transmembrane protein that pokes out of the membrane. They have the M protein, which is a 3 helices of a protein that are inserted into the membrane. And they have the E protein. And they also package the viral genome. So this is what makes an infectious viral particle. Now, thanks to the fact that viral structure and viral genetics are divorced, they're like they're kind of two independent things in virology, which is kind of weird, actually, if you think about it. But thanks to this, you can actually make viral-like particles that in the outside look identical to the infectious particles, but they are not infectious. And so for us physicists, that's a really a nice thing to study, because structurally these particles have all the M proteins, S proteins, of the virus, of the infectious virus, but they're not infectious. So we can have our students play around with them, we can make them, we can ship them. We have been actually shipping a lot of these virus-like particles around the world because they're completely non-infectious and people can use them to study the virus. So I now want to create a case for rapid research on SARS-CoV-2 and say, what could we do? What did we propose? And how did we go about what we are doing? So if you look at the coronaviruses, the common cold coronaviruses, the CoV229E, the CoV-OC43, or the CoV-NL63, they are very seasonably variable. So they all peak at the winter time, and then they ebb during the summer and they peak on the winter time. But if you look at the data really closely, you realize that these viruses actually do not have a given viral seasonability. So each strain is slightly different than the other ones. And it's not given that the virus would be, because it's a coronavirus, it would definitely be seasonably variable. So one of the major questions that popped up when the SARS-CoV-2 virus was identified as the source of the COVID-19 disease was the seasonal variability of the virus. So here is a little timeline. In December, the first, I think it was December 7th, when the first Chinese patient was definitely diagnosed with this virus. On January 7th, the full genome sequence of the Wuhan strain of the virus was published. This is an incredible progress because I think that this is the fastest from disease to viral genome availability that the world has seen. And so rapidly after that, the virus was suspected to be involved in human-to-human transmission. Then on January 30th, WHO declared public health emergency. And so that's here. You see that the total number of coronavirus cases in the world was way low at that time. And then the January 31st, public health emergency was declared in the US. So we are still here. And on March 11th, WHO declared the COVID-19 as a pandemic. And so that's exactly when the number of cases in the world started skyrocketing. This is a very impressive timeline. Meanwhile, at Utah, Michael and I, we were brainstorming over things. And on March 9th, we applied for a rapid award for NSF to study the structural stability of the SARS-CoV-2 and answer this question of seasonal variability, try to answer mechanisms for such a question. And on March 11th, we got funded. So this was a way at the very beginning of the pandemic. And so really the question we wanted to address was when somebody sneezes, they leave behind all of these viruses. And in our case, it was, well, if they're leaving behind viruses, we can replicate that by creating virus-like particles. And the big question was, can we study these virus-like particles? And we can't tell anything about their temperature dependence or seasonal variability. So the first step in this process was to actually make the virus-like particles. So my lab makes a lot of, my lab focuses on HIV. We are very experienced in making HIV virus-like particles. We studied those in great detail, as I will be showing you a little bit earlier. But so within 2 months of getting funded, we were able to create a minimal system for assembly of SARS-CoV-2 virus-like particles. And this system has to do with just expressing these 3 proteins in producer cells. There is the S protein, which is the spike, the M protein, which is the membrane, and the E protein. And these 3 proteins, when you express them, they make a lot of virus-like particles, and you can characterize them with antibodies specific for SARS-CoV-2, and you can see. And you can also take the virus-like particles, incubate them with gold nanobeads that are conjugated with antibodies against the S protein of the virus, and do electron microscopy on them. So these are TEM images. The black bar represents 100 nanometer size. And you can see that the virus-like particles, well, these particles have been dried on the TEM grid. So this is not cryo-electron microscopy. The viruses have been dried, and so they're a little bit collapsed, and they've been stained with heavy metals so that they can be visible in electron microscopy. But you can see that their size distributions is polymorphic. The sizes are in the range of sizes expected for a coronavirus, and we have a lot of the S proteins that are recognized by these gold conjugated with antibodies. So we were pretty confident that we have the virus, we can make virus-like particles, we can purify them, but then can you see a coronavirus? And the answer to that, this is the Argentinian Physical Society, and so I would be blessed if I just give a virology talk. I really need to tell you a little bit about the cool things we have in the lab. So since we have been focused on viruses for a very, very long time, we have really invested in trying to beat the diffraction limit and trying to observe the viral particles. And as we make viral-like particles, we are at the luxury of being able to incorporate fluorescent proteins into these virus-like particles. And so one thing that I wanted to bring up to you is that the interferometric palm measurements have the ability of creating 8 nanometer voxel resolution. So palm, as most of you would have heard by this time, is a method in which you localize single molecules with exceeding precision based on fitting their point spread function using a Gaussian. And so you can localize a single molecule on a glass by a couple of nanometer resolution if you get enough photons. The problem always is the optical axis. So how do you increase your resolution in the optical axis? And so in this experiment that is running on the left, what you're seeing is a very interesting example of increasing the resolution along the optical axis. During this experiment, we have gold nanoparticles that are embedded in glass, and you can visualize them here as these red dots. You would see them at different intensities because the stage in which these glass is embedded is moving up and down only 100 nanometers. So as this experiment is going on, the stage is moving up and down 100 nanometers. But as you can see, when the stage moves up 100 nanometers at the beginning, you don't see any variation in the intensity of one of these beads. So why is that? The reason is that under these conditions, we do not have any interferometric setup. We are really looking at the sum of intensity on 2 cameras, and you're summing the intensity. And although the point spread function of the bead moves a little bit with the 100 nanometer movement, there is not much of a change in the intensity. However, when my student Ipshita actually moves this 3-way beam splitter, that would allow us to compensate for the path in which a single photon travels between these 2 objectives. So I want to make sure you make it clear that this path is really a compensate for a quantum mechanical effect. So every photon that is emitted by one of these beads has a point spread function, has a wave front that goes through both objectives. And this wave front is captured by the objectives and is combined here in this 3-way beam splitter. If the coherence length of that photon is within the distance that these 2 arms have been corrected, then that photon can interfere with itself. And the way we have the prism set up here, you can have it interfere itself in 3 different phases. And so each camera detects one of the phases. And so what you're seeing here in this video is that as this sample is moved up and down 100 nanometers, the prism is moved so that at the latter stage of the experiment, the prism actually is in a place where the interference happens. And then you suddenly see that the photons emitted by these objects actually interfere very heavily by themselves, and they completely disappear from one camera and reappear in another camera. That you can see here. So taking all of these, deciphering all of this information requires a lot of computation power, which we have built in at the University of Utah. And we have done a lot of these studies on HIV. So we have basically looked at the whole lattice of HIV. We have looked inside the HIV. And I don't want to get in there because this has nothing to do with the coronavirus. But I just want to show you a little bit of what we are capable of looking into these small viral cavities and what we can learn from them. So the problem with the coronavirus is that although it's really exciting to see inside the guts of a coronavirus, that would really not answer the question of is there any temperature effects on the coronavirus. But you can ask the question, can you poke a coronavirus? And the answer to that is yes, indeed, you can. You can do atomic force microscopy, and that's, that now you can understand what the collaboration with Michael really brings about, is that you can actually do probe surfaces with atomic force microscopes, and you can have a very flat surface. You can deposit the viral-like particles onto these surfaces, and you can scan them, and you can observe them in atomic force microscopy. Of course, the virus, when you observe it in atomic force microscope, it kind of squishes a little bit, so it becomes a little bit less, the height goes down a little bit, so you're looking at 50 to 180 nanometer virus, whereas the real virus is maybe 150 nanometers tall. So as you're scanning it, the virus kind of squishes down a little bit, but you can observe it. And this was very, very encouraging for us. But the question is, because atomic force microscopy is a topological method, and you're basically looking at surface topologies, can you convince yourself that you actually poke the coronavirus? And this question really took us a long time because we couldn't. Like, it's very hard to convince yourself that 50 nanometer objects scanned by the AFM are actually coronaviruses. So we decided to go back into virology and say, what can we do to make this virus more visible in atomic force microscopy? And so we focused on this S protein, which is the main protein that is giving the coronavirus its name. It's the corona, basically, protein. And we engineered basically an addition to this section of the S protein that would then allow us to attach probes to it. So in particular, we attached a spy tag onto this S protein, and this demonstration basically shows you what we did is that these spy tags were then captured by dead myosins that were also genetically engineered to attach to those spy tags. So when we had the VLPs, we basically incubated them with these dead myosins that had a spy catcher on them. And then these VLPs with a lot of these dead myosins attached to them were really had a high affinity for microtubules. And microtubules are great because they're so easily recognizable in AFM. You have a long structure, you can easily see it. This is really nanoengineering. It's taking some virology and merging it with nanomechanics and nanoengineering to create viral-like particles that like to stick to microtubules. So this was a really, really fun thing to do. And as you can see, we have electron microscopy images of these virus-like particles with the myosins, with the kinesins that are attached to the microtubules. And now we can visualize these in AFM. And so in AFM, when you look at these, this is like little tree branches that have these bumps that we now know are the virus-like particles. So we could then use these to do a structural stability experiment on SARS-CoV-2, because now we had specificity. Now we know that if you have the virus-like particles on the surface, they are the virus-like particles. And this is the manuscript that we just submitted about 2 days ago, in which we used the AFM to look at these virus-like particles that are attached to microtubules on glass surfaces at 22 degrees. And as you can see at 22 degrees in this diagram labeled A, you can very easily distinguish the virus-like particles. But what happens if you deposit exactly the same prep on a 34 degrees stage? So the only thing you have done differently is that you have increased the temperature of the glass that is used in the AFM. And suddenly you see a lot of debris on the glass as your AFM is going around, and you don't recognize the virus-like particles anymore. You can do a control experiment in which you incubate the virus in solution at 34 degrees for an hour, and then deposit it back at a 22 degree surface. And there you can see some of the viral particles, but they're not as destroyed as if this virus was dried at 34 degrees. So basically our data shows that when the virus dries at hot temperature, the structural stability of the virus really becomes it really becomes damaged, and the virus falls apart. So here is, even if you find a virus-like particles at 34 degrees, as soon as you scan it, you basically destroy it. And you can look at these histograms of basically sizes that you detect, because even when you destroy the virus, there is a little leftover left on the surface that you can see with the AFM, and you can see at 22 degrees, you get a very nice peak at around 50, but at 34 degrees, everything kind of collapses, aside from a couple of particles that are still left, but most of the particles are collapsed and destroyed. So what does this technology allow us to do? As you know, the virus is evolving not as fast as other RNA viruses, because coronaviruses have a lot of genetic control, and they actually do not like to make a lot of errors. This comes from the fact that the genome is really large, but the virus is divergent. And so there are these, at every place in the world, there is new strands of the virus coming up. And there is nothing better speaks to this than the biggest mutation that has taken over the world. So this G614 is a mutation within the spike protein that is believed to have increased the infectivity of the virus. So, and that happened somewhere in February in Europe. So the virus that emerged in October is now no longer the dominant virus going around the world. The dominant mutation that happened in Europe is now basically taking over everything. So with our virus-like particle technologies, we are hoping that we can actually catch up and study the virus as it's evolving and kind of look in more detail at the structural stability of the virus. A lot of this research but this research is very difficult, and we have been trying to get data as fast as possible because we are responding to a global emergency. Now, what else can we do with these virus-like particles? This is really kind of a playground for nanomechanics and nanoimaging. We have created, this is of course an image of the virus-like particle that I showed you. The B is an image of fluorescent virus-like particles. So we have managed to tag the M protein of the virus with cherry and also GFP and dendra. And we are now capable of harvesting virus-like particles that are fluorescent. And in a collaboration with Thermo Fisher, we have managed to label the glycans on these S protein viruses with another color. So we can basically create we can probe the sugars exposed on the virus and the viral particle themselves. The image C is what you observed before. This is when we have the virus-like particles attached to the microtubules for AFM studies. And D is a really cool experiment. So in this experiment, we had mouse respiratory mouse lung cells cultured. These have ACE2. And then we added a lot of the virus-like particles, and in 5 minutes, then fixed and embedded these cells into resin. These cells were then thin cut, 80 nanometer cuts, and put in TEM. So you're basically looking at cross-section. What you see on top is the cell, and it's trying to engulf a virus-like particle. And you can barely recognize these S proteins that are basically making connection with the cell. So you can study the internalization of the virus. Remember that from the outside, these virus-like particles look identical to the real infectious virus. So you can look at them being endocytosed using light sheet microscopy. So this is a collaboration with Dr. Kikuchi in Ohio State. He has looked at the ACE2 expressing cells that have basically ACE2 GFP, and he's looking at the virus particles in red, and he can recognize the single viral particles as it's internalized in the cell. So are virus-like particles the end of the road? The answer is no. We are also very, very interested in creating what we call a one-time infectious virus. So in order to study the viral mechanism in more detail, you cannot ignore the replication machinery of the virus. That 30 kilodalton RNA that is 30 kilobases RNA that is embedded in the virus is important for everything that the virus does. So what can we do to replicate that kind of a virus in the lab? We have basically de novo synthesized the whole viral genome of the SARS-CoV-2 minus the S protein. And the reason why we cut out the S protein is because we don't want to create an infectious strand of the virus. We want to create a strand of the virus that is infection defective. And then instead of the S protein of the virus, we embedded the blue fluorescent protein. So these basically, if you transfect these into producer cells, the cells would be capable of creating a bald virus-like particle, which would not be infectious, but it would have the full genome embedded in it. And so by a few substitutions, you can create particles that would have the S protein on these previously bald particles. And you can use these for a lot of different studies. You can use these for how the genome is replicated inside, what's going on. So we are really, really excited to have gotten these cDNAs, because the DNA, the RNA, the genome of the coronaviruses is incredibly difficult to manipulate in the lab, and it suffers through a lot of recombinations and a lot of very weird effects. So these de novo syntheses seem to have basically bypassed a lot of those difficulties, and we are excited to study these particles as they become available. At the end, I would like to just mention that we are expanding. We welcome student and postdoc applications in the lab, both me and Michael. This is a picture of our team. This is the team that created the virus-like particles. This is Michael Vershinin, Abi, Ben, Heather, and myself. And this has been a collaboration with the McKinneys that created the dead kinesin for us and shipped it over. Ipshita Saha is a very talented graduate student focused on the IPALM imaging. A lot of those IPALM papers I showed you were the work of Ipshita, and she focuses on HIV and HIV dynamics. And David Belknap is a really good friend and a collaborator at the University of Utah who runs the EM facility. And so at this, I'm just going to stop and entertain any questions.
Okay, thank you very much. Saveez?
Thank you, Saveez.
You're welcome.
Very, very interesting talk. Thank you. I don't know if anybody in the audience have some question. I was thinking about something that you said, that you shipped to other places in the world these particles you make, or just to do some experiments in another places, or?
So yeah, we ship them because other people want to use them. So we can basically ship the viral-like particles on ice, and other people can utilize them for their research.
Great. So from you've said that temperate is like regarding the influence of temperature and let's say other variables in this in destroying these particles, it's like a combination of temperature but also humidity. What you've said is like the optimal environment for these particles to survive, no?
Yes.
So
we don't have a full picture yet. I want to emphasize that we don't really understand the full picture. We had we basically recreated only a section of the possibilities, which was dry virus-like particles on glass surfaces. Of course, not in every condition the virus is going to be completely dried. Not every surface would have the same chemistry as glass. So there are a lot of different things that we can investigate, but we felt like it's a good point to just take one condition and just show a proof of principle, and then continue on the different conditions, different humidities, and also different genetic backgrounds of the virus, different mutations within the viral proteins.
That's great. And so from this research, you can say something more like general about the influence of climate in these disease?
Yes. So we sent this paper out for rapid communication because we believe that the virus would have the infectivity rate of the virus would dramatically go up as the temperature goes down in the northern hemisphere. It has been a little bit controversial because the virus had, if it was completely seasonable in lock with the season, we would not have experienced a second wave in the summer, which we did experience in the US. But as you know, these things are very complicated, and a lot of places are air conditioned, and so just the fact that the outside temperature goes up, it doesn't mean that the virus does not have any venue to grow. But in the winter time, if the viral particles are more stable at the outside on different surfaces, then one would think that it would be much more difficult to basically they would be much more potent and survive longer.
So the infectivity will increase because
yes, this would argue for an increase in infectivity. Yes, of course. You have to understand that these things are extremely complex, and we are only looking at one subsection of variables. But the structural stability is an important part of virus surviving that dry phase or that droplets phase outside, and so we believe that it's informative.
Okay. Well, one more question is, what about the microtubules that you said that attach or fix the coronavirus? Could be the principle of some medicine or some treatment for the disease?
So the microtubules attachments are good in terms of nanotechnology or biotechnology. So the fact that you can basically create these what we call them the grape-like structures that have the microtubule and a lot of coronavirus on them. This is a great thing to manipulate. So in terms of biotechnology, you can create surfaces in which you have these things. Basically, you can position these things on separate surfaces and then use them to visualize molecular interaction. Like you use them to look at antibody interactions. Like if you can easily deposit these things on glass, and then you can look at how much antibody would basically interact with these virus-like particles. We also have a collaboration with pathology where we aim to inject these virus-like particles into rats and humanized rats and see if the immune response to these virus-like particles is similar to or better than other vaccine candidates. So in principle, virus-like particles are also vaccine candidates. So the combination of things that we can try with these things is quite hard.
So there is no more questions.
Very interesting. I don't see if there are some questions in the. I think no. Okay.
In YouTube, no.
Awesome. Well, it was very nice talking to you guys and being virtually in Argentina. And please feel free to drop me a line and stay in touch.
Thank you so much.